An essential step in the glycolysis pathway is the transformation of glucose 6-phosphate into fructose 6-phosphate. The enzyme phosphoglucose isomerase catalyses an isomerization reaction that takes place during the second phase of glycolysis. Let's examine the glucose 6-phosphate to fructose 6-phosphate conversion in a little more detail. Substrate. The initial step of glycolysis, during which glucose is phosphorylated by the enzymes hexokinase or glucokinase, produces glucose 6-phosphate. The glucose molecule glucose 6-phosphate has been phosphorylated, and the phosphate group is joined to the sixth carbon. Phosphoglucose isomerase. The conversion of glucose 6-phosphate to fructose 6-phosphate is catalyzed by the enzyme phosphoglucose isomerase, also known as glucose phosphate isomerase. PGI is found in the cytoplasm of cells and has a specialized role in the glycolytic conversion of G6P to F6P. Rearranging the carbon backbone of G6P to create fructose 6-phosphate is a component of the isomerization reaction facilitated by phosphoglucose isomerase. The method is best explained as follows. 1. The phosphate group is joined to the glucose molecule 6th carbon in glucose 6-phosphate. 2. By moving the carbonyl group from the glucose molecule 6th carbon to its second carbon, phosphoglucose isomerase catalyzes the conversion. 3. Fructose 6-phosphate, an isomer of glucose 6-phosphate, is created by the rearrangement of the carbon skeleton. The phosphate group is still affixed to the fructose molecule 6th carbon, C6, in F6P. Depending on the energy needs of the cell, phosphoglucose isomerase enables the conversion of glucose 6-phosphate to fructose 6-phosphate and vice versa because the isomerization reaction is reversible. Role in glycolysis. An essential phase of glycolysis is the transformation of G6P to F6P. Following the synthesis of F6P, the succeeding steps of glycolysis continue to metabolize F6P to generate ATP and other metabolites. Why does fructose 6-phosphate need to conform differently from glucose 6-phosphate? In the process of glycolysis, an isomerization reaction transforms glucose 6-phosphate into fructose 6-phosphate. Several factors demand this isomerization. Intermediary metabolism. A product intermediate in the metabolism of glucose is G6P. The metabolic possibilities of the route are increased by the conversion of G6P to F6P. F6P can enter many metabolic pathways and be metabolized to generate energy or as a precursor for other biosynthetic activities. Glycolytic flux. By allowing the glycolysis route to continue, the isomerization of G6P to F6P contributes to the maintenance of the glycolytic flux. The succeeding steps of glycolysis can be carried out quickly and effectively by converting G6P to F6P. F6P is a crucial step that the glycolytic pathway passes through. Regulation of enzymatic activity. The regulation of glycolysis's enzyme activity is impacted by the conversion of G6P to F6P. Several glycolysis enzymes are sensitive to F6P levels. The regulation of F6P concentration, which can affect the activity of downstream enzymes, is made possible by the interconversion of G6P and F6P. Metabolic flexibility. The isomerization of G6P to F6P broadens the metabolic range. Many metabolic processes, including the pentose phosphate pathway, gluconeogenesis, and glycogen production, use F6P. The conversion allows the carbon skeleton to be channeled into multiple metabolic routes dependent on cellular energy demands and metabolic requirements. Carbon rearrangement. The carbon skeleton must be rearranged in order for G6P to become F6P. The ensuing processes in glycolysis and other metabolic pathways depend on this rearrangement. It enables the production of many intermediate products and the effective utilization of the glucose molecule. In general, the efficient development of glycolysis, metabolic flexibility, control of enzyme activity, and utilization of glucose in diverse metabolic pathways depend on the conversion of G6P to F6P by isomerization. It ensures that the carbon skeleton of glucose is channeled efficiently into the right metabolic pathways to meet the cell's energy and biosynthetic demands.